Vint Cerf, father of the Internet.
DNA is the blueprint that defines every living system on the planet, not just human beings. So it's fundamental to life. And now we have a method for reading and interpreting this information at scale.
It's absolutely amazing when you start to think about the application, especially in some of it's medical related too, as you start to think about mapping DNA, of millions of people against and just even solving viruses and things like that. Never would have been thought to have been able to be done, and now you can do that, right? Which is
well, actually, just picking on the DNA and the virus question. Of course, the purpose of DNA is to produce proteins,
yep,
and enzymes and things like that, signaling proteins and so on. And the configuration of the DNA and the various methylation and so on determines what gets expressed and what doesn't get expressed. The question is, what do those proteins look like? And something just simply extraordinary was reported out of DeepMind, which is one of the alphabet companies in the UK. They were able to get their protein folding program based on deep neural network learning to essentially represent, in a way that they believe is accurate, something like 88% of all the proteins that could be generated or are generated by human DNA. So, I mean, this is an astonishing accomplishment done with extremely clever machine learning techniques and with the help of a human population that ran the AlphaFold program on their laptops. Now, you remember SETI at home, the search for extraterrestrial intelligence, was pulling in all these radio signals, and then they needed to be analyzed to see if there was any regularity to them. And the way they did it was to allow people to run the SETI at home application in the background on their laptops. They'd suck in a little chunk of digitized received signal, and then they'd analyze it. And of course, you were supposed to raise your hand if you found something interesting. They did something like that with AlphaFold and allowed people to help train the algorithm to correctly get the folding sequence to work right. So they made use of this machine learning and the assistance of people to train the algorithm. So it may not just be quantum computing that produces exciting results. It may also be this new form of computation called deep neural networks.
It's astounding what we're coming to in the next couple of years, and it doesn't sound like we're that far away from really sea change ability to solve problems that have confounded us for decades, right?
While supercomputing had benefited, and actually ordinary computing had benefited from the reduced instruction set computing idea, which Dave Patterson and John Hennessy pioneered. And got the Turing Award for not too long ago. That reduced instruction set allowed, simplification of the instruction set and increasing in clock speeds, increasing numbers of cores to produce increasing amounts of compute cycles available for computation. But what's been happening is suddenly a proliferation of heterogeneous computing. So the conventional CPUs are now joined by graphical processing units, or GPUs, which run like hell and were originally intended just to draw triangles as fast as you could to do graphics. But now we've repurposed them to do special kinds of integer computations at blazingly high speeds. Then we have the tensor processing units, which are configured to do multi-layer neural networking at blazing speeds. And then, of course, the potential quantum computing coming along. So now just imagine for a moment that instead of having this uniform gigantic pile of RISC processors, what you actually have is this mix of, I even left out floating point, field programmable gate arrays, FPGAs, is another element of heterogeneity. So imagine that you've got all these different kinds of computational capability. And your job is to design a programming language and a compiler that figures out how best to apply all that heterogeneous computing to solve this problem.
Yeah.
And, holy moly, it's a whole new headache. So in some wonderful sense, every time we think we've finished, somebody comes along and says, no, you haven't.
The new coronavirus genome looks like this. It's just under 30,000 bases long. The coronavirus is made of a type of genetic material called RNA. But scientists convert it to the more recognizable DNA to make it easier to study. That's what you're seeing here.
With all of the challenges when we walk out the door of our house right now with COVID and all this stuff, it's still amazing that technology is marching on at a very, very fast pace in the background. I mean, just switching over to, obviously you've been watching as a technologist, this whole vaccine that got kind of turned out in a matter of months. To move a vaccine through and get it out the way it happened, I think was pretty amazing. I mean, I don't know I'd love to hear your perspective.
Well, several things came together to allow this rapid development. First of all, the ability to sequence the genome of the virus itself quickly is something relative, which is developed over time, and it's now possible for them to do that sequencing very, very quickly. So that's the first thing. The sequences came out within a couple of months of the discovery of this virus, and they were made available over the internet very, very quickly. So people who were interested in understanding how the virus worked from the point of view of the protein structure of the virus, and especially the spikes, because that's how the virus injects its RNA into a cell. So we had the sequence very, very quickly. We had tools that helped us understand what the shape of that spike or the spikes are, and we could figure out how it was that they were able to interface to the ACE2 receptor. And of course, the next step is how to figure out how to screw that up. And so finding ways to inhibit the ability of the virus to actually connect to a receptor so it's unable to squirt its mRNA or RNA into the cell and hijack the cell's protein production. So all of those tools were available at a time when we needed them and would not have been available, certainly not in the previous big pandemic. So we had tools available, and we had the net available in order to exchange information. And of course, there was enormous amounts of collaboration that took place. The side benefit of the net, of course, is that it allowed a lot of people to continue working, even though they could not be in proximity to each other, and they could do what we're doing right now. Not everyone's work permits that. And so we saw supply chains breaking. We saw a lot of businesses like bars and restaurants having to shut down because they couldn't serve people in proximity to each other. So there is a great asymmetry to the impact of the pandemic on various livelihoods. And now, of course, we're at the point where we're trying to get everybody vaccinated. I am astonished that there are people who reject the vaccine on the grounds that somehow or other it hasn't been adequately tested. I think there are probably 500 million injections have happened since we began making those vaccines available. I don't know how much bigger a test you want in terms of eliciting any potential hazardous outcomes or risky outcomes. In any case, we really need to get people vaccinated, because the more people who are not vaccinated, the higher the probability they will contract the disease, and the higher probability we will see new mutations happening, because it's the replication of the virus that creates the mutations. And so the less it propagates, the less mutation we have. The more it propagates, the more mutation we have, and the higher the probability that some of those mutations will be worse than the already terrible Delta variant. So I'm hoping that not only will people come to their senses, but that the vaccine will become more broadly available everywhere around the world. We desperately need to get it into the hands or into the arms of people who haven't been vaccinated yet.